for the time being, we'll move the epigenetic module to a later part of the module. So we want to introduce you to some cancer-related resources. So we mentioned the TCGA has profiled 10,000 tumors, and there are places where you can either download the raw data or the processed data. You can also write a program to interact with the data source. These are called the Genomic Data Common, and Broad Institute and University of Chicago both have a Genomic Data Common for the TCGA data. And these processed data are still kind of in big text file format, whereas Sloan Kettering and recently together with Dana-Farber, we're kind of like co-developing this CBio portal, which has really just point and click, all the processed data you can visualize, you can see the figures, you can look at which samples are differentially expressed or have some mutations. You can play with it. And they have initially just TCGA data, but recently they are collecting more and more public expression data or mutation data now. UC Santa Cruz so has this Xena function, which also has API. So yeah, your GUI is you have to point and click, and API, you can actually write a program to directly interact this to access raw and processed cancer data. You can also use it for some analysis. Many years ago, University of Chicago, Michigan has this OncoMine. You can also look at processed public data for visualization. This one has more microarray data. We haven't followed up to see whether it has all the new sequencing data. Whereas Sanger has this Cosmic, which has, they annotate those recurrently mutated cancer genes. And you can see if you're interested in a gene, interested in one gene, it can show you what is known about all the mutations on this gene in cancers. And you can see whether one base is really recurrently mutated, how often the mutation happens as missense, or truncating, and so on. So these are very, very useful. So in summary, there are many, many different profiling approaches that are used by the TCGA and other cancer profiling consortium. People are starting to look at different subtypes. It could be based on mutations, could be based on expression or protein level. Later on, we will also explain that you can look at the different subtypes based on DNA methylation. And interestingly, some of these DNA methylation subtypes also co-occur with some mutations as well. And depending on the different subtypes or mutations or expression clusters, we can see that they can have really different survival. And we talk about Kaplan-Meier survival curve and how you can use Cox regression to look at what different genes or factors influence the patient survival in those subgroups. And you can also later on use survival analysis to see whether a particular drug can help the patient. And in order to really figure out, whether the gene is a oncogene or tumor suppressor, or whether it's a gain of function or loss of function, we usually look at the location of the mutation, whether it's amplification or deletion, whether the expression level also indicates that if it's a higher expression or lower expression of gene, and all of this together helping us to understand whether we are seeing a loss of function mutation on a tumor suppressor or a gain of function mutation on oncogene. And one biggest surprise people found was mutation on epigenetics. And we'll talk about that later. You have a question, Jed? No? Okay. Great. Yeah, so we'll just finish now. Okay.